morning, friends. So now we are going to start the new topic, and that is systemic pathology. So whenever we are going to study the systemic pathology topics, so each and every bacteria we are going to cover, and we are going to study detail about that pathogenic agent or that bacteria. So in general, the points which we are going to cover in systemic pathology is the history of that organism, then the morphological features, cultural characteristics, the biochemical reactions, resistance of that organism, then the antigenic structure or virulence factors of that bacteria, then the laboratory diagnosis, the pathogenicity of that bacteria, laboratory diagnosis, epidemiology of that bacteria, and in few bacteria, if immunoprophylaxis is there, that also we are going to cover. And the final is the treatment part. So treatment of this infection caused by this organism. So from systemic bacteriology, the first and the most important topic for your both examination, theory as well as practical, is a staphylococci. So staphylococci, these are gram-positive cocci, occur in grape-like clusters. They are indicators, means they cause the different type of infection, cutaneous infection, respiratory tract infection, systemic infections. And that's why they are called as a ubiquitous in nature. First time these cocci observed by von Recklinson in 1887, and in 1880, another scientist, that is Louis Pasteur, isolated this bacteria from pus. And after that, the Sir Alexander Austin proved the pathogenicity or pathogenic effect are produced by this organism. The pyogenic infection, pyogenic means pus-forming infections, are because of the infections of Staphylococcus, and that is discovered by Sir Alexander Austin, a Scottish surgeon, in 1880, the same year. Then the the meaning of Staphyli, also he gives the name Staphylococci. Alexander Austin gives the name Staphylococci. Staphyli means bunch of grape. They are arranged in a bunch of grape, cluster arrangement, that is called as. And coccus means berry. So another scientist, Robert Rosenberg, isolate two species of this bacteria, Staphylococcus aureus and Staphylococcus albus. Aureus means gold. So aureus produce golden yellow color pigment or nuclear agar, and Staphylococcus albus produce white color pigment after going on to the nuclear agar plate. In the same year, in 1885, so this Rosenberg in 1884 he discovered these two species, and in next year in 1885 another scientist Pasteur discovered one more species from the skin, and that one is Staphylococcus citrus. So that is called as the Staphylococcus citrus, which produce lemon yellow color colonies. So aureus, golden color, albus, white color colonies, and citrus produce lemon yellow color colonies. In early days, we are using this pigment formation characteristic of Staphylococci for the differentiation, for the classification. So, but nowadays we are classifying Staphylococci by using the coagulase test. Coagulase positive Staphylococci and coagulase negative Staphylococci. So this genus is having near about 32 different species. So more than 32 different species are there and 15 subspecies this one is having. But out of these 32, only few are medically important. And the remaining these are not medically too much important. So this one is a picture of Staphylococci. So during the binary fission, it divides in different plane. The cell division occur in different plane. That's why such type of cluster type of arrangement occur. Bunch, they are arranged like a grape of bunch. Looks like a bunch of grapes, and that is called as a cluster type of arrangement. Then for the classification of these cocci nowadays, we are using the coagulase test. So depends on the coagulase positive and coagulase negative, we are differentiating the bacteria, this Staphylococcus aureus. So only one example of coagulase positive, and that one is Staphylococcus aureus. Well, the another example of the different species of Staphylococcus aureus, they are coagulase negative and medically important. These are the Staphylococcus epidermidis, Staphylococcus saprophyticus, Staphylococcus hominis, and Staphylococcus captis. All these are the coagulase, CNS, coagulase negative staphylococci we are calling it. So these are the pictures under the electron microscope, and this one is under the routine our oil immersion lens, another violet color. So you may also observe this under a compound microscope under 100x oil immersion lens. They are arranged in a cluster. Cluster arrangement is a very important characteristic of staphylococci. So this one is again another picture. So you observe this. So indirectly in the smear. So from directly in the smear, so we are observing like this. They are arranged in the clusters. Now we'll start the Staphylococcus aureus. Out of these, the most important and the most pathogenic species is a Staphylococcus aureus. So aureus means which produce golden. The meaning of aureus itself is a gold. Gold means aureus. So which produce golden color pigment, golden color colonies of the surface of nutrient agar. That's why it is called as a aureus. So morphology, as usual, the first point is morphology. These are spherical, 1 micron in diameter. As these are the foci, we are measuring the size in diameter. Grape-like clusters. So they are arranged in the clusters. These are non-motile and non-spore forming. Non-motile means flagella is absent and spore forming activity is also absent. That's why these are non-sporing. Readily stained with aniline dyes, uniformly gram positive. They are violet in color after staining with gram staining, and that's why they are gram positive. Grow on or cultural characteristic. Now in cultural characteristic, grow on ordinary media. These are simple. These are simple organisms. They are non-fastidious in nature. That's why grow on any ordinary media like simple media, nutrient agar. After growing onto the nutrient agar, temperature required is near about 10 to 42 degrees centigrade. pH is 7.4 to 7.6. These are arrows. Always they grow in presence of oxygen, or sometimes may grow in absence of oxygen. So facultative in arrow or facultative in arrow. Majority medically important bacteria they are from this group, aerobic or facultative anaerobic in nature. After growing onto the solid media like the nutrient agar, colonies develop after overnight incubation after 24 hours, and the colonies are 2 to 4 millimeter in diameter, circular, convex, smooth, shiny, opaque. Opaque means non-transparent and easily emulsive fiber. Most strains produce golden yellow pigment. So the most very important characteristic of this is golden yellow pigment. Formation of golden yellow colonies onto the nutrient surface of nutrient agar is a very important characteristic. So golden yellow pigment. Nutrient agar, if you grow onto the nutrient agar slant. Slant means the media in a tube. It shows oil paint appearance. Again, this one is very important characteristic. Oil paint appearance is only shown by S aureus after growing on nutrient agar slant. After growing onto the blood agar, hemolysis seen. It shows beta and alpha hemolysis particularly. Then on McConke's agar, after growing onto the McConke's agar, pink small colonies, pink color, small pinpoint colonies are produced by this organism, and that is because of lactose fermentation. After lactose fermentation, acid is produced. Neutral red indicator is present in McConke's agar, and in acidic condition, neutral red is pink color. That's why pink color colonies are produced by Staphylococcus aureus or any lactose fermenting bacteria. So this one is LF, lactose fermenter, pink color. After growing into the liquid media, uniform turbidity generally occurs in liquid media. So these are the pictures. Now you see the golden color exact. So white color by albus and golden color by S aureus. Again, you see the difference between alpha hemolysis and beta hemolysis. These are beta hemolytic. Now the biochemical reactions ferment many sugars, but the most important is mannitol fermentation. Always it depends on this mannitol fermentation. Catalase test positive, urease test positive, reduce nitrate to nitrite means nitrate test positive, indol test negative, methyl red and August Prosker MRVP positive. So These are very important biochemical. Always for the identification of bacteria up to the species level, it depends on biochemical reaction. Study of biochemical reaction is must. So this is about the biochemical reactions of Staphylococcus aureus. Now, catalase test. So very easy test it is. You also in last lecture in identification we complete. The reagent we are using is H2O2. Hydrogen peroxide. Take a drop of H2O2 onto the slide, pick up the growth colony with another slide and just dip. Air bubbling indicates positive test. What happens? An enzyme catalase is produced by this organism. It split H2O2 into H2O and inert oxygen. And that's why air bubbles are produced. So these are positive control, negative controls. Indole test negative. So indole test is negative. Now the resistance of this organism, this vegetative cell withstands 60 degree centigrade for 30 minutes. It remains as it is for 60 degree for half an hour. Some require 80 degree for 1 hour to be killed. So resist phenol for 15 minutes, resistant to lysozymes, sensitive to lysostaphine, where sensitive to penicillin. Penicillin resistance, nowadays we are observing, and here three type of resistance are noted or studied. Production of beta-lactamase. An enzyme beta-lactamase is produced by this bacteria. The gene which is responsible for the beta-lactamase production is present in plasmid. What is plasmid? Plasmid is extra chromogenic material. So extra chromogenic material is present, and the gene which is required for the production of beta-lactamase is also present in that plasmid. So which synthesize the beta-lactamase acts on beta-lactam ring penicillin, and because of that, penicillin group of antibiotics become non-functional, unuseful. Then change in bacterial structures receptor. The receptors, penicillin binding receptors are there. So for penicillin group of antibiotic, a particular receptors are there that binds, antibiotic binds to the receptor and it shows its action. But nowadays the changes occurs. It may be a phenotypic or genotypic because of that such changes occurs, and that is because of that also the resistance, generated resistance occurs. Development of tolerance also develop, and because of that the resistance against the beta-lactamase group of antibiotic or penicillin group of antibiotic generally occurs. Pathogenicity and virulence produce two types of disease: infections and intoxication. Infection because of direct involvement of that bacteria, and intoxication because of the toxin produced by this bacteria. Few complications occur, and that is called as an intoxication. Toxins produced either in the host or performed in vitro. Directly producing the host or in vitro means outside the body. Means inside the food or like that food poisoning. Now the epidemiology, yes or yes, is frequently carried by healthy individuals on the skin and mucous membranes. Carriers serve as source of infection to themselves and others. For example, by direct contact, by contamination of vomits, food, which can result in food poisoning. Pathogenicity and virulence, cocci, damaged skin or mucosa, added to the cell, multiplication occurs, tissue damage and infection. So, in infection, such type of cycle occur, cocci attached to the skin, damaged skin or mucosa, added to the cell, multiplication occur, tissue damage and infection occur. Post-formation cells associated polymers. So this virulence factor, what are the different virulence factor? Cell associated polymers, cell wall polysaccharide, then teichoic acid and capsular polysaccharide. Cell surface proteins, extracellular enzymes and toxins. All these are the virulence factor. Virulence factor, they increase the pathogenicity, they increase the virulence of this bacteria. Cell-associated virulence factors we see. So capsule or slime layer, glycocalyx, peptidoglycan, teichoic acid is conventionally linked to peptidoglycan and is species specific. Yes or yes is having rhabitol teichoic acid, while Staphylococcus epidermidis is having lacetol teichoic. There are two types of teichoic acid, acetol and rhabitol and lacetol. Then protein A is conventionally linked to peptidoglycan. That one is also act as virulence factor, clumping factor, or bond coagulation. Then teichoic acid. Now, cell-associated polymers, the teichoic acid. So, it is an antigenic component of the cell wall, facilitates adhesion of cocci to the host surface and protects from opsonization. So, how it increases the pathogenicity? How it increases the virulence? First, it acts as an adhesion, firm attachment to the host cell. Firm adhesion, attachment of this cocci to the host cell. And also it protects the opsonization. Then capsular polysaccharide, whenever the fresh isolates are always capsulated, surrounds the cell wall, inhibit opsonization, and inhibit phagocytosis as well as inhibit opsonization. Protein A, these are also present at the outer side of the cocci, major component of the cell wall, binds to FC portion, the IgG. So IgG antibody is having FAB portion and FC portion, and it attaches to the FC portion of IgG, and FAB portion remain free for the attachment of another antigen. And such type of coagulation, such type of agglutination is known as coagglutination. So this we are using for coagglutination test. Includes platelet damage and hypersensitivity. Also, protein A is responsible for this hypersensitivity and platelet damage. Clumping factor, also called bound coagulase, responsible for slide coagulation test. So bound coagulase, there are two types of bound coagulase and free coagulase. So clumping factor is a bound coagulase and responsible for slide coagulation test. This organism also produces enzymes, and they are also responsible for pathogenicity, to increase the pathogenicity of this organism. And these are the coagulase, the lipase, hyaluronidase. So these enzymes are responsible for to increase the pathogenicity. So coagulase, mainly produced by SRES, brings about clotting of human rabbit plasma, acts along with the coagulase reacting factor CRF. There are 8 types have been identified, most human strains from type A. The difference between free coagulase and bound coagulase. So coagulase, free coagulase, and this clumping factor are bound coagulase. Secreted into the medium freely, required CRF for its action. Required coagulation reacting factor for action. So constituted to the cell wall, does not require CRF for its action. Action is independent. Enzyme, lipase. So produced by coagulase positive and negative staphylococci, acts on lipid present on the surface of the skin, capable of destroying the tissue and may facilitate the spread of infection to adjoining tissues. So this lipase enzyme is responsible for spreading the infection from one side to another side. Now the most important is the hyaluronidase, produces by many strains of SRES, hydrolysis hyaluronic acid, permitting the easy spread of infection. So from one side to another side, in connected tissues, this hyaluronic acid is present, and it breaks down the hyaluronic acid because of the hyaluronidase, and spread is easily possible from one side to another side. It's spread. Then it produces few toxins, and these are also responsible to increase the pathogenicity of this bacteria. So these are cytolytic toxins, enterotoxin, toxic shock syndrome toxin, and exploitative toxin. So such 4 different types of toxins are produced by this organism. They increase the pathogenicity. Cytolytic toxins, membrane active substance, contains 4 hemolysins and 1 leukocidin. Hemolysins, alpha, beta, gamma, and delta hemolysis. Alpha hemolysis is the most important. Lyses erythrocytes, damage platelet, macrophages, cause severe tissue damage. Leukocidin, it produces the leukocidin, and that one is responsible for exotoxin lethal to PMNLS contributes of the invasion of the organisms by suppressing phagocytosis. So it inhibits phagocytosis in short. Then enterotoxin, so a very powerful enterotoxin produced by this organism which is responsible for food poisoning. So responsible for manifestations of staphylococcal food poisoning, heat stable exotoxin, a type of identified A to E and GI. Produces by 30 to 50% of Staphylococcus aureus isolates. Now the next one is toxic shock syndrome toxin. Classic cause of TSS, sometimes referred to as Staphylococcus enterotoxin F, described by James Todd in 1978, rare but potentially fatal multisystem disease characterized by high fever, hypotension, shock, associated with the use of highly absorbent vaginal capsules in menstruating women. Exploitive toxin. So one more very important toxin produced by this organism, and that one is exploitive toxin. Or also it is known as scalded skin syndrome toxin. Scalded skin syndrome toxin, also known as epidermolytic toxin, and causes scalded skin syndrome in children, causes the epidermal layer of the skin to slough off. Scalded skin syndrome, also known as writer's disease, the disease also being recognized in adults. So you see the picture, so such type of picture occur, the upper layer of epidermis is separated, and all over the skin system of the regions separately occur. That is because of the scalded skin syndrome toxin. Now the pathogenicity of this organism. So in pathogenicity, clinical disease, staphylococcal pyogenic infections or the cutaneous infection. It produces cutaneous infection, respiratory tract infection, and different systemic infections. So we'll start one by one. So staphylococcal pyogenic infections in cutaneous, it causes the folliculitis, furuncle, carbuncles, bullous, impetigo, paneritia, and pyrorhizia, wound infections, mastitis, osteomyelitis, staphylococcal pneumonia, all generally caused by this organism. Jadhav said, Jadhav, ते काय त्यात रामग्रेड असतात. Different staphylococcal diseases range from localized to systemic. So localized cutaneous infection. So it causes the localized cutaneous infections, invade skin through the wounds, follicles or glands. Folliculitis, superficial inflammation of the hair follicles, usually resolves with no complications but can progress. Furuncles, while inflammation of hair follicles or sebaceous gland progresses into the abscess of the tissue. Carbuncles occurs under the skin, so that means large and deeper lesions created by the aggregation and interconnection of the cluster of these furuncles. Impetigo, bubble-like swelling that can break and peels away, most common in newborn. So clinical diseases, staphylococcal intoxication, staphylococcal food poisoning, exploitive intoxication, writer's disease or staphylococcal skin syndrome. Staphylococcal scalded skin syndrome. Staphylococcal toxic shock syndrome also produced by this organism. So these are the few pictures. Boils or peduncles, styes to the eyelid, generally system of infection occur. These are surgical wound infections, may cause including S or S. These are the first forming small lesions occurs onto the skin. Again, these are the different type of skin. Yes. Then it also produces musculoskeletal infections like osteomyelitis, arthritis, or bursitis, bone infection. These are. It also able to cause respiratory tract infection in which the tonsillitis, pharyngitis, sinusitis, otitis, bronchopneumonia, and lung abscess. Sometimes it may cause the CNS, central nervous system infections, abscess, meningitis, intracerebral thrombophlebitis. Also, it is responsible to cause endovascular infections like the bacteremia, septicemia, pyemia, and endocarditis or pericarditis. Then the staphylococcal disease epidemiology, pre-reservoir for staph or intra-antigen regions of mucous membrane, then transmission, direct contact, and inanimate objects. HIV infected patients have significant increased carrier. So it is more dangerous in HIV infections. The laboratory diagnosis is the most important. After this pathogenicity, lab diagnosis, specimen collection, microscopic examination, and cultures. In laboratory diagnosis of Staphylococci, specimen collection, pus, sputum, blood, feces, vomits, urine, nasal, and perineal swab, and remaining contaminated food also we are using in food poisoning. Direct smell examination, plenty of pus cells with gram-positive cocci in cluster, but cannot be differentiated between the different species of the Staphylococci. So such type of picture of gram staining, gram-positive cocci, and in clusters. These are the already we learned this spherical one. Then the culture we have to inoculate onto the nutrient agar plate, blood agar plate, McConkie plate, and a few selective media also we are using where the chances of contamination. So nutrient agar, blood agar, McConkie medium, mannitol. Now these are the selective medias: salt, mannitol agar, mannitol salt agar, then Ludlam's media. Other selective media: milk agar, glycetol, acetate agar, or Ludlam's media. So these are the pictures from this golden yellow color cornea produced with hemolysis. So biochemical reactions, coagulase test is very important. Coagulase positive, ferment many sugar including the mannitol. Mannitol is important. Catalase positive, urease positive, reduced nitrate test positive, phosphatase production positive, MRVP positive, indole negative. Catalase test already we explained. So what happens is H2O is break down into H2O and inner toxin because of that. Immediate depressions occur. This one is a catalase test. Slide coagulase test. Bond coagulase, also known as clumping factor, is detected by using this. This coagulase is bound to the bacterial cell wall, hence it is named. It reacts with the fibrinogen in plasma of visible clumps. So take a drop of plasma and mix. Take a drop of this suspension, mix. Clumping occur. So such type of clumps occur. Positive test. Here the another one is negative test. Tube coagulase test, free coagulase released by the bacterial cell. It exists as a free form, unlike the bond coagulase and attached to the cell. The free coagulase acts on prothrombin, producing a complex staphylococci. This substance then acts on fibrinogen to form a fibrin clot in the plasma. So the same, that is 0.5 ml rabbit or human plasma we are taking, equal amount we are mixing. Incubate at 35 for 4 hours, check after clot formation. You till the tube. If the test is negative, it will flow. If the test is positive, clotting occurs. It doesn't flow. So like this. Then coagulase. This one is no vaccine susceptibility. Also we are using to check the zone of inhibition. Then penicillin resistance. For to check the penicillin resistance, production of beta lactamase. So plasmid mediated enzymes will inactivate penicillin by splitting beta lactam. Staphylococci produce 4 types of penicillin, ABCD, all these. So change in bacterial surface receptor. We already studied this. Protein binding, reduction in protein binding proteins. Development of tolerance. Staphylococci develop the tolerance to the penicillin, are only inhibit but not kill. Methicillin MRSA, now the very important methicillin resistance is methicillin resistant Staphylococcus aureus. Nowadays they are causing the hospitalized infection, secondary infection. If the patient is admitted in the hospital, these organisms are present in the surrounding. They cause the infection. These are a very serious type of infections. And for treating these infection, nowadays we are using the antibiotics, that is linezolid or linezolid. Generally we are using, or ticoplanin we are using. Risk factors, okay, diabetes, intravenous drug, all these are generally the risk factor. HIV, AIDS, cancer, transplant, osteoporosis, young children, they are older. So all this comes under the risk factors. This is called by MRSA, sepsis, toxic shock syndrome, necrotizing facet pneumonia, deep pus-filled boils and cellulitis, respiratory tract infection, open wound infection, intravenous catheters infection, urinary tract infection, necrotizing fasciitis and polymyxis, infective endocarditis and bone and joint infection. All these are because of MRSA, methicillin resistant Staphylococcus aureus. Mainly two type of MRSA, community acquired MRSA and hospital acquired MRSA. Detection of MRSA, phenotypic methods and genotypic method, oxacillin agar screen method and real-time PCR. So nowadays we are using this for detection. So this oxacillin, then this community associated MRSA, utramexazole, tetracycline, doxycycline and clindamycin treatment. And hospital acquired vancomycin, ticoplanin, linezolid and daptomycin we are generally used for the treating such type of patients. The coagulase negative staphylococci, important agents of hospital acquired infection, prostate devices and catheters. There are only two most important. These are the S. epidermidis and S. saprophyticus. Staphylococcus epidermidis and saprophyticus. Staphylococcus epidermidis, normal flora of the skin, common cause of the infection of the implants and such as heart valve and catheters, common cause of stitches abscess. Staphylococcus saprophyticus, it is also generally present onto the skin and it is an opportunistic pathogen. So normal flora of the skin and periurethral area, frequent cause cystitis, tends to be sensitive to most antibiotics, no acid resistance from epidermidis, Staphylococcus epidermidis. That one is sensitive and this one is resistance. Thank you. Then this topic is over.